Hello, everyone. Thank you for joining us for our November Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Huntington's Disease Foundation. To start, I want to thank our sponsors for this webinar, Ionis Pharmaceuticals, Novartis, Bristol Myers Squibb, Neurocrine Biosciences, and Unicure. And a big thanks to everyone who supports the HDF. It is truly because of you that we are able to fund exciting science that is transforming what we know about Huntington's disease. And of course, a big thank you all for being here. I'm happy to introduce everyone to 3 HDF-supported young investigators. Young investigators are truly the future of Huntington's disease research. They are the ones that are spending countless hours at the lab bench, making the discoveries that are shaping what we know about HD. They are also the future leaders of the top HD labs around the world. So today you will hear from 3 of these people who will each share their work for which they received HDF funding. We are joined today by Dr. Xiaojing Sui, Dr. Icnelia Huerta Ocampo, and Dr. Hassan Fakih. Dr. Sui has studied HD throughout her career using a multidisciplinary approach to uncover how proteome conformational instability drives aging and neurodegenerative disease. Her work aims to define the molecular principles of proteome maintenance and identify strategies to restore proteostasis and resilience in aging and disease. She was recently recognized as a rising star in proteomics and metabolomics by the American Chemical Society for her innovative contributions to quantitative structural proteomics. Dr. Ocampo in the Davidson Lab works on neurodegenerative diseases using a translational approach. Her current research focuses on developing gene therapy strategies aimed at improving treatment options for disorders of the basal ganglia, including Huntington's. She is passionate about advancing therapies that can ultimately improve the quality of life for people within families affected by these diseases. And Dr. Fakih focuses on enhancing the delivery of siRNA therapeutics. He employs self-assembled nucleic acid particles and lipophilic conjugation strategies to precisely control protein binding and siRNA pharmacokinetics. Ultimately, Dr. Fakih aims to develop an effective siRNA-based therapeutic for various diseases and CNS disorders. Before I pass it over to Xiaojing, I want to remind you all that you can ask questions in the Q&A box or the talk. So try to remember to use the Q&A box, not the chat box. We'll hopefully have about 15 minutes at the end for Q&A. So type your questions in as you think of those, and we will try to get to as many of those as we can by the end. Also, a reminder that this will be recorded and available on our website and YouTube channel in about 2 weeks. So if you want to watch it again, or if you want to share it with someone who you think would enjoy it, it will be available for that. Thanks again for everyone for being here, and I will pass it over to Xiaojing.
Thanks, Sarah, for the very kind introduction. Hello, everyone. I'm Xiaojing Sui. I'm a HDF fellow at Northwestern University. Today, I'm very excited to share my research with you, watching proteins change their shape in Huntington disease, and how this knowledge may inform us for the future therapeutic strategies for Huntington disease. I grew up in a historic town in northern China. This great stone bridge is one of the oldest surviving stone bridge, built over 1400 years ago. As a child, I was amazed by this structure because it showed how human creativity can really overcome the challenges posed by nature. That thinking inspired me to pursue a career in science. My research journey began at Chinese Academy of Science, where in Professor Jiazhen's lab, I had the freedom to explore chemistry and biology and build tools to study Alzheimer's disease. This really lays the foundation for my multidisciplinary research with a focus on big data. I then moved to University of Melbourne in Australia and started my PhD with Danny Hatters and Gavin Reid, where I developed a deep interest in protein aggregation in Huntington disease. You know exactly what protein aggregation is because you must have seen this in the kitchen. When you fry an egg, the proteins in the egg white will absorb heat and form these white protein clumps. A similar process happens in the brain of Huntington disease patients. After studying these aggregates extensively, a logical question to ask is, what happens before these aggregates formation? This is important because understanding this process will allow scientists to design therapeutic approaches to delay or even prevent these aggregates formation. This leads me to Northwestern and started my postdoc with Professor Rick Marmotto, who is a world-class leader in aging and neurodegenerative disease. In Marmotto lab, we use C. elegans to study Huntington disease. So what is C. elegans? Sidney Brenner has a brilliant answer for this. It's nature's gift to science. Sidney has this unique vision to bring this 1 millimeter long roundworm to the spotlight of biological research. Over the years, discoveries using C. elegans has led to 4 Nobel Prizes. The first one was awarded to Sidney Brenner, Bob Howells, and John Swanson. By the way, Bob is also a founding member of the Scientific Advisory Board of HDF for their discoveries on genetic regulation of organ development and programmed cell death. You may be very familiar with the second Nobel Prize. It's to Andy Fire and Craig Mellon for their discovery of RNA interference. This is the foundation for many of the current gene therapies seen in Huntington disease, as well as in other genetic disease. It reminds us how powerful basic science is to transform how we treat human disease. Now let's come back to Huntington disease. Huntington disease is caused by the CAG repeat in Huntington gene. When it's less than 35, it's non-pathogenic. When it's more than 35, it's pathogenic. We can visualize these repeats in C. elegans. As you can see here, CAG19 formed this soluble protein as marked by green in C. elegans, and the animal move freely. However, when the CAG repeats increase, you can see they form these large protein clumps. The animal moves really slowly. So in C. elegans, we can really recapitulate these protein aggregates as well as the toxicity seen in Huntington disease. So now come back to the question I asked at the beginning. What happens before these protein clumps form? Imagine a protein as an origami. It has to fold into its correct shape. When it doesn't, we call it a protein misfolding, which is very prone to form these toxic protein clumps, as commonly seen in aging and neurodegenerative disease. We humans are made of millions of cells, and each cell has thousands of proteins. So in this very crowded environment, it is very challenging for proteins to fold properly. They need the help of quality control network, including chaperones, which promote proper folding. Meanwhile, the misfolded proteins can be quickly removed by degradation machinery. However, when the cells express mutant Huntington, they are highly prone to aggregation. During this process, they will sequester these quality control machineries from their normal housekeeping pathway and leaving less efficient quality control machineries for other cellular proteins. Therefore, leads to the buildup of misfolded proteins. So now the question is, when mutant Huntington misfold and aggregates, how many other proteins misfold? To answer this question, we first have to solve a very challenging technical problem to separate these different folding states. This is where my project comes in. We developed a highly sensitive approach to watch protein misfolding. Here's how it works. We use a molecular scissor to cut proteins into small pieces, and different shapes will generate different pieces. These can be identified and distinguished by an advanced machine called mass spectrometry. Then, after a series of computational analysis, we can really watch protein misfolding. The power of this technique is it allows us to watch thousands of proteins at once. Therefore, I can really ask this ambitious question: when mutant Huntington misfold, how thousands of proteins in our cell change their shape. We apply this approach in C. elegans. Strikingly, we find that mutant Huntington misfolding has domino effects on many other proteins in C. elegans. This suggests that Huntington disease is not just as a single gene disorder, but as a collapse of a protein homeostasis affecting many cellular processes. In other words, mutant Huntington misfolding is just the tip of the iceberg. So now the next big question is, how do we prevent this from happening? Let's revisit protein misfolding pathway. You already know that our cells have these built-in cleanup machines to remove this misfolded protein. One of the machine is called proteasome. The beauty with the proteasome is that it can selectively target these misfolded proteins, not the well-folded proteins. And then it works like a shredder to cut those proteins into smaller pieces and remove the misfolded proteins from our cells. And thanks to HDF funding, now we are studying how proteasomes are impaired in Huntington disease. Once we figure out exactly which part of a proteasome go wrong in Huntington disease, we hope to engineer hyperactive proteasome that has enhancing activity. Therefore, they will be more efficiently cut these misfolded Huntington as well as misfolded other proteins in the cell. This could lead to the reduced level of mutant Huntington at both its misfolded version as well as aggregated version to restore protein homeostasis. This is still work in progress, but we are very hopeful about what we are going to discover. To make a summary, what I show you is misfolded Huntington is just the tip of iceberg. It has this collateral damage on many other proteins in our cells. We aim to boost proteasome activity, therefore to remove many of these misfolded proteins and to restore their cellular function. With that, I would like to thank people in Muramoto lab and for lots of scientific discussion, as well as for the C. elegans work. The mass spectrometry work was done in collaboration with Prado, Finley, and Gigi lab. And the proteasome work was done in collaboration with the He lab and the Finley lab. Also thank Northwestern facility for providing the platform for my big data analysis. Special thanks to Huntington Disease Foundation to support our work, which traditional funding agency might find too ambitious. With that, I'd also thank you for your listening. With that, I would like to pass it to Dr. Icnelia.
Hello, everyone. I would like to thank the Huntington's Disease Foundation for inviting me to speak today. This is really a fantastic opportunity to present our work and share it with this community. And I'm very grateful for the support we receive from you, which makes this research possible. Before talking about the projects that we are doing in the lab, I would like to introduce myself and share with you a little bit about the journey I have followed and that has taken me here. My name is Icnelia, and I come from Mexico City. I originally trained as a medical doctor, but very early in my career, I became fascinated by the brain, knowing how it works and what happens when something goes wrong. That led me to pursue a career in neuroscience research. I earned both my MD degree and my PhD degree from the National University of Mexico, UNAM. Soon after, I took a plane that took me to England, where I continued my research work at the University of Oxford. When I was boarding that plane, I just thought I would stay there for a couple of years, never imagined that it would turn in nearly 12 years, during which I built a wonderful life and formed a lovely family. As a postdoctoral researcher, I joined the lab of Professor Paul Bolam, a leader in the basal ganglia research field. It was there that I discovered the microworld of neurons, their anatomy, connectivity, and how do they connect to other regions in the brain. Soon I became deeply interested in knowing how these circuits support such important functions like movement control, cognitive process, and decision-making. Also, by studying these circuits, it taught me something important. To understand what goes wrong during disease, we first need to know how these circuits are supposed to function in health. And then we can begin to unravel what goes wrong in basal ganglia brain diseases like Huntington's disease. After many years of being in Oxford, I came to the US and I joined Rutgers University in New Jersey, where I continue doing basal ganglia research work in animal models of Parkinson's disease. Nevertheless, I wanted to give the next step, which was not only understanding what goes wrong in brain disease, but also exploring how we might be able to correct it. That let me join the lab where I work now, which focuses on designing gene therapies to treat neurodegenerative diseases. The Davidson Lab is based at the Children's Hospital in Philadelphia and is led by Beverly Davidson. We are on the 11th floor of the Colquitt Translational Research Building. Don't know yet. Got the stock. I'm sorry. All right. Our lab studies genetic conditions that affect the brain, including Huntington's disease. We explore why some subtype of neurons are more vulnerable than others. Using advanced tools in animal models, we can follow how this disease progresses over time and how different cells respond. For me, having trained as a medical doctor was important to bridge science and medicine. I would love to believe that the research we do in the lab as a team can eventually reach the clinic and make difference in patients' lives. It's this connection between understanding disease mechanisms and developing therapeutic strategies what keeps motivating my work. Many of you might be already familiar with Huntington's disease and what causes it, but I would like to focus on key aspects of the disease that our lab studies and how we're developing strategies to potentially slow its progression. Huntington's disease is caused by an excessive number of CAG repeats in the Huntington gene. This leads to the production of a harmful form of the protein, Huntington, which accumulates and gradually damages neurons. Some groups of neurons are more vulnerable than others, especially those located within a region of the basal ganglia called the striatum. However, the striatum is strongly connected to the cerebral cortex, another brain region that it's strongly affecting in Huntington's disease. Within the striatum, we find the main type of neuron, which is called the medium spiny neuron. They received this name because they're covered, all the dendritic branches are covered by multiple tiny spines. Medium spiny neurons in the cortex, in the striatum, receive dense cortical innervation by pyramidal neurons, which cell bodies lie in the cortex. These neurons send the projections and the axons make synapses and contact these spiny striatal neurons. This circuit is crucial for controlling movement, cognitive process, and these functions are heavily disrupted as disease progresses. Now, one of the earliest changes in Huntington's disease occurs at the level of gene regulation. Normally, neurons have a very balanced system that regulates which genes should be turned off, which ones should be turned on, and the intensity or the level of expression of these genes. In Huntington's disease, this balance is lost and becomes increasingly disturbed over time. Gene activity starts to shift in abnormal ways. So genes that should be on and should be expressed highly, they are not, and vice versa. This growing imbalance that we call gene dysregulation becomes more severe as the disease progresses. So in our research, we're testing whether changing and correcting the two major drivers of Huntington's disease and lower by two different strategies, Huntington and slowing as well the CAG expansion, we can balance and cells can regain this normal gene activity. So this takes us to our first experimental strategy, which is if we lower the amount of Huntington, can we restore these disrupted gene patterns back toward normal? So to study potential treatments, we use mouse models of Huntington's disease that develop many of the same features we see in HD patients. Very clever people in the lab design gene therapies using harmless AAV viruses that can safely deliver treatment to neurons. Our goal is to treat the most vulnerable neurons, which are located in the striatum and the cortex. So for this, we deliver our treatment straight into the striatum. However, as we know, as I just explained, the striatum is heavily connected to the cortex. In the striatum, we have many axons that belong to pyramidal neurons in the cortex. These axons are able to uptake our treatment and transport it back through its axon to the cell bodies that lie in the cortex. So by doing this, we can treat medium spiny neurons and cells, neurons in general, in the striatum and neurons in the cortex at the same time. So in order to get to our final goal, we need to go through two steps. In the first step, we have tested the effectiveness of our HTT lowering treatment in the short and long term. We have done and performed dose response experiments that help us to choose the right dose, which produces the most potent effect without having toxicity. We have validated the efficacy of our treatment using a wide range of molecular tools. And by doing so, we have found that the treatment was able to reduce both the mRNA messenger for Huntington and the levels of Huntington protein itself in both the structures, the striatum and the cortex. Now, with this information, we have started to move into the next phase. In our second step, we will be conducting single-cell sequencing experiments and depth analysis of these experiments using single-cell tools. This will allow us to study thousands of individual neurons one by one. And with this approach, we will be able to answer very important questions. We know in anticipation that not all neurons will respond the same. But this approach will help us to answer, for example, which neurons received the treatment, showing in green, which ones did not. And from the ones that received the treatment, do these neurons show an improvement in the abnormal gene expression patterns? So are we able somehow to rescue or help these vulnerable neurons to get healthier? Now, our lab also focused on some other aspects of the disease. We're interested in knowing what happens to the CAG repeat in Huntington's over time. We know that CAG repeat is not stable, and it can keep expanding in the neurons over time in a process called somatic expansion, which makes mutant Huntington protein becomes more harmful and accelerates disease progression. So as a second strategy, we have designed therapies to slow this expanding process. So if we do this, can we restore this gene dysregulation in these neurons and bring it back toward normal? So normally our cells have repair systems, DNA repair proteins that can fix DNA abnormalities or mistakes. However, in HD, some of these DNA repair proteins cause the CAG repeats keep expanding. So we have asked, if we reduce activity of these proteins in relevant neurons, can we slow or stop this expansion process? And if by doing so, can we restore the dysregulated gene patterns back to normal? For doing this, we have followed the same approach using a different RNA-based therapy specific for these proteins to lower down the expression of these DNA repair proteins using as well various models of HD mice, doing the same delivery strategy and targeting both structures of neurons at the same time. We're following the same 2 steps that will lead us to our final goal. As a first step, using the same kind of tools, we have been able to show that our treatment reduces the mRNA for the repair protein, the total levels of the repair protein itself. Our treatment was able to reduce the number of CAG repeats, as well as a number of mutant Huntington aggregates in the striatum. These results are very encouraging, and soon we will be moving to step number 2, which is following the same strategy as I just explained before. So just as an overview, I've presented to you 2 different RNA-based therapies. We have done and we have gone through the molecular validation by doing several experiments. We have confirmed the efficacy of the treatment from both the strategies, HTT lowering and slowing CAG expansion. And we're ready to move to the next step, where we will be doing the single cell sequencing and see how different neurons respond to this treatment, and if we are able to correct this disrupted gene expression. I want to thank the Huntington's Disease Foundation for the support they have given us, and that makes this possible. I would like to thank also the Davidson Lab, especially people mentioned here, and to you for listening. Thank you so much.
Hello, everyone. I'm glad to be presenting today. Thank you so much to the HDF for giving me the opportunity to present some of the work we're doing here at the RNA Therapeutic Institute at UMass Chan Medical School. And the title of my talk today is going to be Nucleic Acids from Central Dogma to Therapeutics. So before we start with the science, I would like to give you a little bit of a background about myself. So I come from this tiny dot on the map called Lebanon, and that's actually where I conducted my studies, my undergraduate studies at the American University of Beirut. So this is the beautiful campus here at the Mediterranean Sea. And I was a chemistry pre-medical student. So I actually wanted to become a pre-medical doctor so I can help treat patients with various diseases. But during the course of my studies, I was doing some research with this professor, Professor Pierre Karam at the time. And this is where I was really hooked on research and chemical development, because it was the first time I was introduced to how science can help us think outside of the box, where I specifically was developing DNA-based sensors that can detect viral infection. This has then led me to drop out of medical school and then move very, very far from Lebanon to Canada, where I did my PhD studies in chemistry at McGill University. And I continued on the same path. I wanted to explore more what we can do with DNA. So I worked under the supervision of Professor Hanari Sleeman, where her famous quote always in my head is, take DNA out of its biological context. So in my PhD, what I was working on is how can we improve drug delivery? So that's the delivery of drugs, make them more effective and take them to the right spot at the right time. But we were actually using DNA as a nanotechnology material. So we're actually using DNA, if you think about it, like Lego pieces and piecing them into different structures that can allow us to make these drug delivery materials purely from DNA. But then I wanted to do a little bit more translational work, and I got really interested in RNAi therapeutics, or small interfering RNAs, which led me then to move from Canada to the US, where I am currently positioned now. And I'm a postdoctoral fellow in Anastasia Korova's lab, who has pioneered chemically modified siRNAs. And I'm currently working on developing RNA therapeutics for various diseases. But what is this talk really about? So this talk is really about nucleic acid therapeutics and how can we use nucleic acids not just as DNA and RNA material, but actually as therapeutics themselves. So nucleic acids, we have two different flavors of them in our cells. We have deoxyribonucleic acid, or DNA for short, and we also have ribonucleic acid, or RNA for short. And all I want you to remember from this slide is that these are using a 4-code system. So we always have A, T, C, and G in DNA, and A always binds to T, and C always binds to G. And the RNA, we have the same exact code, except instead of T, we have a U. That's all I need you to remember for today. So what is the central dogma, and how does disease happen actually? So the central dogma of life starts with, we have our DNA in the cell that encodes our genes. It gets transcribed into what we call a messenger mRNA. And then this messenger RNA gets then translated into a protein, which then goes on to do various functions inside of our body. Think of it as the DNA is like the book that encodes commands, which are the genes. The messenger RNA is the messenger delivering these commands to the cell. And then the proteins are the performers of these commands. Now you can imagine if something goes wrong with this system, if the proteins are altered, maybe there's too much of a certain protein, or there's too little of a certain protein, or maybe there's some misfolding happening into this protein, it's not actually functioning as it's supposed to be, then we have the healthy state or healthy function lost, and this is usually how disease happens. And typically for us as humans, the way that we develop therapeutics, we have two classes of therapeutics that are very common to most of you. We have traditional small molecule drugs. So these are very small chemical drugs, such as Tylenol that we take for our headaches. Or we have biologics, or most commonly antibodies. So this, you can think about it such as Humira, which is used for autoimmune diseases. And usually these therapeutics come and bind to these disease proteins and prevent them from causing disease. But you can see there are some challenges with these therapeutic modalities. Even if these therapeutics are developed, sometimes we cannot find a pocket for them to bind on the disease protein. Additionally, not all proteins we want to target might necessarily have a pocket at all. And most importantly, even if we bind the disease protein and preventing it from making disease, you can imagine if the disease is genetic in nature, then the disease protein is always going to be made. So this is where kind of the term came into existence, the non-druggable targets. But then scientists started thinking, well, what if we target the genetic basis of these diseases, the source of the error, if it exists, either on the RNA level or on the DNA level? And this is where nucleic acids again come into the picture. As I just remind you that DNA and RNA, they use this 4-code system where A will always bind T or U and G will always bind C. So a groundbreaking discovery happened in 1978 by Paul Zamechnik, where he was able to synthesize in the lab a short DNA strand encoded here in red, which we call an oligonucleotide, that's shown here in this picture. And he designed this oligonucleotide to be complementary to this mRNA shown in blue, which was causing infection inside of cells. When he was able to bind this mRNA, which is causing the infection, he was able to prevent the infection from happening by preventing the viral protein from being made. So he effectively blocked the translation using an oligonucleotide, or a DNA strand. And this is where the name, or the coining of the antisense oligonucleotide came into existence. And again, I remind you, this can be designed simply by knowing the sequence of your target. So in this case, if the mRNA virus has UUU, you can design your ASO to be AAA, and then it will bind it, and it will prevent it from being translated. And as you can imagine, this is a revolutionary discovery because now we were able to use nucleic acids themselves as therapies that can alter the gene expression. We can prevent it from happening. And this led to the explosion of the field over the past 30 or 40 years, where we have now what we call nucleic acid therapeutics as a new class of therapeutics made only from nucleic acids. And we have different kinds of nucleic acid therapeutics that I've shown you here. We have antisense oligonucleotides, we have aptamers, small interfering RNAs, CRISPR-Cas system, and even mRNA, which most of us have received in the form of vaccines. What's unique about this class of therapeutics, which are NATs, is that they allow us to control gene expression at all levels. So if I bring back here the central dogma, you can see that the CRISPR, for example, can work at the transcription level. ASOs, siRNAs can block translation, but mRNA can increase translation. And aptamers can even be designed to bind the protein themselves. So you really can cover this whole pathway using nucleic acid therapeutics. And in fact, to date, we have more than 20 approved nucleic acid therapeutic drugs. And what's unique about them, they are very selective and specific because they're based on the complementarity of the sequence. In my particular research, I focus on one class, which is small interfering RNA, or siRNA for short. And what these molecules do, they allow you to lower the gene expression. So what you do is you degrade the mRNA of interest and prevent the protein from being made. And a typical siRNA drug looks like this, where you have your siRNA sequence here, and you have a chemistry or a delivery ligand enhancer that allows this siRNA molecule to perform really well inside of our body. And just to give you an example, this is clinical data from patients, where here these patients received an siRNA treatment. The ones in yellow were the control patients. They did not receive any treatment, and they have a steady expression of this protein. But then when they receive a single injection at day 1, you can see this protein is now decreased by about 80%, and this decrease is actually lasting up to 240 days. So this is really unique to siRNA therapeutics that give you very long-lasting therapeutic effect. You can imagine going to the doctor once a year to get a shot, and that's what we're hoping to develop. So now, where does nucleic acid therapeutics come into play with genetic diseases such as Huntington's disease? My colleagues have presented to you Huntington's disease, and I'm pretty sure you guys are very familiar with it. It's caused by the mutant Huntington gene that encodes more than 37 CAG repeats, and we currently don't have any treatments available. And then someone might ask the question, well, why don't we develop siRNA therapeutic against the mutant Huntington and just treat the disease? And the question is, why aren't we there yet? Well, the reason for that is because we have two major challenges with siRNAs for Huntington's disease. The first one is the siRNA delivery challenge, and the second one is the complex nature of Huntington's disease. So the siRNA delivery challenge, you can imagine we want the siRNA to be stable in the body, and we also want it to be delivered to the brain, which is a little bit difficult, but not just deliver it to the brain. We need it to reach the right region in the brain, which is mostly the striatum that's mostly affected. And also inside of the cell, we need it to reach the cytoplasm of the cell, where it can actually perform its function. And then even if we can get over these challenges, Huntington disease over the past few years, we've seen that it's actually more complex than we thought. The mutant Huntington indeed induces aggregation on toxicity, but now there are other targets that are implicated in the disease. And these include mismatch repair pathway, as you've heard. There's also inflammation. So this really necessitates for us to take a more holistic approach in treating Huntington's disease, or what we want to do in our research is to achieve multi-targeting of these problematic issues. And this is where my research is focused on. We want to use chemistry to address these issues, obviously because I have a chemical background, right? So in our lab, in the Korova lab, what we've seen is that if you have an siRNA molecule shown here on the top, we can add these different chemical modifications, and we can stabilize this molecule using different chemistries. So these are stabilizing chemistries, allows the molecule to be stable, safe, and be delivered well inside of the body. But then what we observed is that if you take two siRNA molecules and you link them together, this gives you a huge boost of delivery. So when we inject this divalent siRNA structure that has two siRNAs into the mouse brain, and then you look at the red signal, which is the siRNA drug, you can see that it covers the end of the brain to the front of the brain, including the regions that are impacted by Huntington's disease, which are the deep brain regions. The other nice thing about this molecule is that you can imagine you can design one siRNA sequence to go against one target and then the other sequence to against another target. And that allows you to have dual targeting approaches where you can treat two different genes that are causing Huntington disease pathology. So overall, this improves stability, delivery, as well as allows us to target two genes at once. So where is my research focus right now? The focus is how can we develop multi-targeting siRNAs? And I'm trying to really answer two important questions. When we linked these two siRNAs together, it performed much better than a single one. And we're trying to understand, is the size responsible for this improved delivery in the brain? And then the second question is, can we actually target more than just two genes? Can we go beyond that to address, as I mentioned, HD more holistically? So the molecules that I'm trying to synthesize in the lab are either targeting 3 targets at once or 4 targets at once. And you can see that these can actually be very powerful molecules that we would really like to test in the context of Huntington's disease. Obviously, this is a little bit ambitious, and I would like to give a shout out to the Huntington's Disease Foundation, who supported this work through a postdoctoral fellowship. And we're really making really good progress on this end. So just kind of to summarize, what is the big picture of this research? We want to understand how can we improve drug delivery to the brain and the ultimate goal to maximize the therapeutic effect. We also want to have the ability to have multi-targeting disease entities so we can address all aspects of Huntington's disease. So for example, if you look at this molecule, target 1 could be targeting aggregation. Target 2 is targeting mutant Huntington. Target 3 is for somatic expansion, and target 4 could be for inflammation. And then last but not least, we would hope that this drug delivery modality would be employed also beyond Huntington's disease, because this is modular. We can simply change the targets we're going after and apply it for other disease-causing genes. So I'll just leave you with one last data figure in this presentation just to show you where we are right now. On this bar graph, each gene is represented by a color. So you have gene 1, gene 2, G3, and G4. And on the left is the control. So these are mice that are not injected with any siRNAs. And then on the right is our multi-targeting component. So you can see we're able to lower all 4 genes simultaneously with a single drug. So we're really excited. We really believe in this technology, and we hope that we're able to advance it in the future as a therapeutic modality for Huntington's disease. With that, I would like to shout out the people who helped me in this work, specifically every member of the Korova Lab, my mentors, Anastasia, Hanadi, and Masad, and the HDF Foundation. And I'm happy to pass the mic back to Sarah, and happy to address any questions if there's any time.
Thank you all so much for sharing your work. I love it because it's 3 totally separate strategies, right? And I think it goes to show that in HD research, we don't have all our eggs in one basket. There are people that are taking very, very different approaches to look at HD. So I think it's really exciting. Okay, so we will kick off the questions. So we have some questions that are already in, but make sure you enter your questions into the Q&A box as we continue our Q&A session so that we can get to your questions. All right, so I think I will start with one for Icnelia. They're asking, given that neurons may die rapidly once CAG repeats reach about 150, it seems the therapeutic window for Huntington lowering approaches could be relatively narrow compared with strategies that target somatic expansion. Could you comment on this and share your thoughts on the ideal timing or therapeutic window for Huntington lowering interventions?
Yes. Well, it's important because as disease progresses, everything becomes more complicated. The mutant Huntington starts to build up and starts to really damage neurons. So we need to intervene at a step where we can really make these changes. We are doing our experiments in young mice, so that will be equivalent to a stage of the disease that it's not so developed. So so we hope that intervening at the right window will help to prevent all the cascade of events that happen. But of course, the windows are very important in disease progression, and we really need to target in the right timing.
Thanks, Icnelia. So here's one for Xiaojing. If we could control the aggregates, could we treat Huntington's disease?
Yeah, that's a very good question. That is what we are working on in Marmoto lab and also in collaboration with our other labs. The misfolded and aggregated protein of mutant Huntington or the other proteins caused by mutant Huntington misfolding, those are the driver for the proteome toxicity, and also the proteins, they lose their function. So if we can target all of them and then lower down the misfolded version of those proteins, we can really to restore their cellular function. I think that's one direction we really want to focus on.
That's exciting. Thanks. Okay, so this is a kind of a two-part question. I'm going to group it to a two-part for you, Hassan. So the first question is, what were the 4 genes you targeted with the tetravalent siRNA? And then the second question is about the bivalent RNA. They ask, have you tested it? What if the second target is not present? How does it affect binding?
Yeah, excellent question. So in the data that I've showed, the genes that we've targeted are model genes. So one of them is mutant Huntington. The other one was JAK1, so it's involved in inflammation. But the other 2 targets were just model genes. They're not necessarily directed at Huntington's disease genes, but we do have genes that we can target. So for example, we're thinking about a combination of potentially somatic repeat expansion targets such as MSH3, Huntington, and then inflammation. So that's kind of what we're thinking. But really, it's the power of the system is not necessarily the biology, but really the modularity. So you can apply this to really any gene we think is implicated in Huntington's disease and do an assessment if it has an impact on the pathology. In terms of the second question, if the second arm doesn't target anything, we still believe it will aid in the distribution in the brain. We believe that having a bigger size molecule allows it to distribute really well and deliver to different brain regions. So you can design it to not engage any gene necessarily, but you will still benefit from having it there to increase the size of the molecule.
Thanks, Hassan. So maybe this is one, so it came in for Hassan, but I think it might be good for Icnelia as well. So maybe Icnelia, you could kick it off. They want to know about for gene silencing or lowering. Is there any more information on downstream impacts, such as symptom changes? So is there any changes with symptom outcome? It's great to see that you're showing efficacy in lowering gene expression, but it would be great if we could see there's improved pathology, of course. So do you want to maybe talk about that, Icnelia? Then we'll kick it over to Hassan.
Yeah. Well, yeah. Well, the lowering strategies always come with some hiccups, right? So, for example, in just talking, I'm going to talk about the 2 different lowering strategies we are following. So by lowering Huntington, we're also lowering Huntington itself, not just the mutant version of it. So that comes with lowering Huntington as a wild type, as a protein that helps also in a good way for neurons to communicate and make synapses. So that's one of the consequences that we're not specific in lowering mutant Huntington itself. So that can have that could impact in a not in a bad way, but it also has this downside effect, right? But with lowering the repair proteins, we are those combined. We hope that we have a synergistic effect, and we are actually trying to combine these 2 strategies and have both lowering therapies at the same time. So I forgot the second part of the question. Sorry.
No worries. It was, I think you hit on it. It was about symptom control.
Yes. So some of what we have seen is by lowering these repair proteins, we have been able to reduce the mutant aggregation, which is one of the pathological hallmarks of Huntington's. So we think that by reducing this mutant Huntington aggregation, then we're hoping that we are extending the health of the neurons, and this doesn't become so toxic and pathogenic. So we could be able to slow disease progression and somehow slow the neurodegeneration.
Thanks, Icnelia. Do you want to add anything, Hassan?
I'll just add that at least from our work, we haven't really tested yet the impact on downstream in these combination treatments, but we do have recently, we've put out a preprint from the lab. We're showing that targeting, for example, mutant Huntington and the repair pathway does synergize to alleviate the symptoms observed and the changes in the RNA and stuff in the brain. So we do believe that this moving forward that this is possible, and we do expect to see symptom changes when we approach it in a more holistic manner.
Thanks, Hassan. So I have a question for you, Xiaojing. So obviously Huntington's isn't the only disease where we see these protein clumps. Could you talk a bit about advancements that have been made in studying proteostasis for other diseases where you see kind of these aggregates form?
Yeah, I think, our strategy looking at the proteome is also kind of trying to not only hit Huntington disease, but also Alzheimer's disease, Parkinson's disease, ALS, which also feature by this aggregates formation, right? Those protein toxicity. And there was a paper from David Smith's lab. They really engineered these hyperactive proteosomes, and they can have better degradation, efficiency for different A beta. And I think that they test A beta and other mutant proteins involved in the neurodegenerative disease. So I think it's really promising that, if we target this, our cell built-in degradation machinery that can clean up specifically this misfolded protein or non-functional proteins, that is one of the way to go.
Thanks. And so I'm aware of the time, so I think maybe this will be a good question to end on. And I think it kind of brings in why so many people stay in the HD field, and that's the personal nature of this. And so someone asks, this may be a naive question, feel free to ignore. My 30-year-old daughter was just diagnosed with HD, a 41-mer. Can you hint at the likelihood of a cure in 10 years, 20? Again, there may be other resources for answering this. So maybe we could just kind of go around and give your thoughts. Obviously, no one has a timeline on this, but I think, when people are new to this field, it kind of just helps hearing from researchers what their thoughts are. You want to start, Icnelia?
Well, I think Huntington's disease research has been changing a lot throughout the years. And I think there is a lot of new innovative strategies, different strategies. So what we presented today, that we can cover the disease from different aspects using now more advanced tools that are available nowadays. I think there is and with this community that really enhances collaboration. We share our discoveries and we can build upon them. So I think we're moving in the right direction. I can see that this will be advanced and will be moving forward year by year. So I'm really hopeful that this will change how we treat the disease, and not just symptomatically, but from the roots and the causes, we will be able to modify it.
Thanks, Icnelia. Do you want to add anything, Xiaojing?
Yeah, I think, at my presentation at the beginning, I showed how basic science can really transform. We treat human disease, and I think understanding all of these, all of the rules of this disease and why it's happening, and also to understand the wide effects of this gene, and really will help us to understand the mechanisms and finding out the different treatment approaches towards the same goal. I'm also thinking like Hassan's approach using the different multiple model, and there's also a possibility to use siRNA to target negative regulator proteasome that can enhance the proteasome activity. So there are many possibilities, and we work very hard in the lab and trying those ideas, and we are hopeful.
Thanks, Xiaojing. Hassan, do you want to add anything?
I'll just echo the hope. I have the same hope as my fellows have mentioned. I think I obviously it's hard to put a date or a year on it, but I think science has been advancing exponentially, and I think everything is possible. We're doing a lot of things that we didn't think we can do. So I think there is hope for sure, and I think bringing people to the table from different backgrounds, such as what the HF is doing in terms of basic science, all the way into clinical advancements, is the right way to do it. So we cannot give you a date, but I think all of us here are working very hard towards this. So.
Thanks. Yeah, and I mean it's so tough, right? Because I feel like that's what so many families want when they first get an HD diagnosis is when are we going to have a treatment in hand? And so I guess I would add, I don't think, I don't think we're going to have a cure in 10 years or 20. I think cure is kind of a dangerous word, but treatment, I think it's very likely that we're going to have one in hand in 10 years. And I know for families it can feel like we're taking 2 steps forward, 2 steps back, but science is an iterative process, and particularly with the new Unicure news, right? I know that was a big blow for people with the reversal of the FDA decision. And so I think to that, I would say that science is iterative. We have a lot of eggs in different baskets, which I think our researchers here highlighted so nicely today. And I think, when we look at the future, it's going to be kind of, we have to find what works first and then we can improve upon it. And I think we're very, very close to being there. And so I would say if you are dismayed by the recent FDA decision, the HDF has a petition out on change.org. You could find it on our website. We just dropped a link into the chat. Please feel free to add your signature to that. That is going directly to the powers that be at the FDA to try and change that decision, because we do want to see these treatments move forward as quickly as possible. So I am aware that we are directly at 1 o'clock. So I'm going to wrap it up there. If you had a question that we didn't get to, I'll send those over to our speakers. So if you left your contact information, they can get back to you. I want to end by again thanking our sponsors, Ionis Pharmaceuticals, Novartis, Bristol Myers Squibb, Neurocrine Biosciences, and Unicure, as well as everyone who donates to the HDF to support the research that we fund. And of course, everyone for tuning in. And last, but certainly not least, our speakers today, Xiaojing, Icnelia, Hassan, thank you so much for taking the time to share your research with everyone today.
Thank you.
This is our last webinar of 2025, but we are going to be kicking things off again in January for our 2026 lineup. I will be the first speaker of our 2026 series to recap the highlights from 2025, and between what we've learned from basic research as well as clinical trials, there's going to be a lot to cover, so you definitely won't want to miss it. Make sure you're on our mailing list so you can get emails about registration for that webinar as well as our 2026 lineup once it comes out. We are so proud to showcase the advancements in HD research through our Research Spotlight webinar series. So if you find these sessions valuable and you would like to support our mission, please consider donating today. Thank you for being a part of our community, and I hope you all have very happy and safe holidays, and I will see you in 2026. Thanks so much, everyone.